Lucy, you're from Cambridge and Sanger Institute. Tell me exactly what you're doing there, because your position in the Sanger is what, for instance?
I've just finished doing a PhD in the lab of Peter Campbell. I'm a clinician scientist, so I'm a clinical oncologist in my full-time job, and I took 4 years out to go and do a PhD in the cancer genomics lab at the Sanger Institute.
Right, so you're a scientist and a doctor, and
yes.
You're looking at cancer, you're looking at relapsed breast cancer. Why were you doing this project, looking at the genome and looking for factors to indicate the possibility of relapse?
So we know that the very root cause of cancer is defects that develop within the genome of once normal cells, and they transform into cancer cells over time as a consequence of these defects in the genome. So what we wanted to know is how cancers will relapse in the future. What is it that makes one cancer curable and another cancer incurable and come back in the future?
Of course, classically, we look at the primary, do some tests and say, yes, we can treat it in this way or that way. But this might change.
Yeah, so these findings suggest that even if we see something in the primary tumour, that might not necessarily be the same thing later on. We find that often there are additional things have actually happened within the cancer genome over time.
What have you found that has changed?
So we've found by looking at individual cancers that new cancer genes are sometimes found at relapse that weren't seen in the primary tumour. And we find there's a wide range of cancer genes that can be involved in that situation. And this really gives us a challenge when it comes to precision medicine, because we're seeing a very wide range of genes can be implicated late in evolution.
Right, so there is this evolution going on, but how can you anticipate that and look for clues so that you could potentially target some of those late changes?
So we compared the genomic landscapes of primary and relapse cancers. So we looked at 839 cancers where the primary tumour had previously been sequenced. This is what we know the genomic landscape looks like. And then we looked at 161 relapse cancers, and we tried to find differences between them. And some of those differences we see suggest that there are certain subgroups of primary cancers that seem to be more likely to relapse, and that fits with a lot of previous research looking at gene expression. So that's one way that we find that potentially, if we can isolate that group, that would really help us to prevent the relapses at a later stage.
Have you seen any potential therapeutic targets at this stage?
So in terms of therapeutic targets, we can look at those in the primary tumour or those in the relapsed tumour, or those shared by both of them. And actually the majority of mutations that are targetable are actually present in the primary tumour. So there is hope for targeted therapies at that upfront time. And this contrasts to some other cancers where a lot of the events are late that you may target. So actually a lot of the events are in the primary tumour. And then the question is, these additional events that we see arising late, are they so clinically important we need to be targeting them as well? And we will work that out over time by doing more of these sequential sampling approaches. What
do you advise busy cancer doctors to be doing now about all of this very exciting potential?
I think as an oncology community as a whole, we need to start thinking about how we can keep up to speed with the sequencing technologies. Sequencing technologies are allowing us to sequence and look at a base pair level every single gene in a patient's cancer. Okay, we need to get lots and lots of patients' cancers to look at to meet this scope. And we need to do that over time. We need to see how people's cancers are changing over time. So we need to start thinking about how we can sample cancers at different points in a patient's journey.
And you do genome-wide sampling, and this could supplement the classical pathological science.
It's only going to be by getting big enough numbers that we're going to be able to break down the complex landscapes we see and start to make sense of them to bring real improvements for individuals in the future. What
do you think is the interim take-home message for cancer doctors then?
I think cancer doctors and clinical trials need to start sampling patients' cancers more frequently, banking tissue so it's available for the technology to work on, and that's how we're going to bring real improvements for patients.